And there we go. All right, so today we're going to cover how we study microbes, okay, which is covered in chapter 1. So here is an overview of what we're going to cover today. We're going to cover this week's learning objectives, and kind of it's my way of letting you know how what we're covering today works toward meeting those learning objectives for the course. I'll take any questions you'll have. And then we'll talk about the scientific method, Koch's postulates, microscopy, staining microbes, which will be highly useful for lab, and then what to do for Monday of next week. All right, so here are this week's learning objectives. So last time we talked about evolution. We'll be talking about how populations have changed to result in the populations of bacteria that we have now. Identifying microbial structures and connecting the structures to their functions. When we talk about staining and that kind of thing, we'll be talking about structures. And then identify pivotal components of microbial systems important to human health. We'll be talking about pathogens, which are important to human health. And we're going to analyze and describe the impact of microorganisms, mainly with pathogens. Okay. All right, any questions? Any questions from your reading? Any questions about Blackboard, assignments? Okay, nothing at this point, but if you have questions as we go along, feel free to let me know and I'll stop and I'll be happy to answer your questions. All right. Okay, the scientific method. So first off, let's talk about what the scientific method is. Okay? The scientific method is a set of steps that we take to answer a testable question. Okay, so okay, what is a, well, let's back up. I'm not quite ready for that yet. So a testable question is something that we can set up an experiment to answer. Okay, now there are lots of questions that we have a hard time answering with the scientific method. Okay, so later on we're going to see how Dr. Robert Koch, back in the 1800s, used the scientific method to determine that Mycobacterium tuberculosis caused tuberculosis, and that's why they named it that. So it's a rod-shaped acid-fast bacterium that causes the disease tuberculosis. So he was able to definitively show that that happens. When we get into other areas that are more in the realm of the metaphysical, they're harder to answer with the scientific method. For example, the nature of consciousness. We have been struggling with that one for eons, ever since humans achieved self-awareness. And it's really hard to answer with the scientific method. You know, we're giving it a shot, but it's really hard to get definitive answers to that one. Okay, so that is what the scientific method is. And it helps us determine the difference between correlation and causation. Okay, so one thing that's important to keep in mind with the scientific method is we start with observations. Okay, we see something like, hey, people who live together in the same home tend to all get tuberculosis. Now it's really easy to jump to a conclusion and say, okay, it must be the house. That's something that we could test. Okay, and that is something that they did test. Okay, another conclusion that was come to, that which actually, you know, given the evidence, was that the first person to die of tuberculosis comes back as a ghost and sucks the life out of their family members because they're lonely and they want their family members to be with them on the other side. In other words, you know, vampires. Now that may seem silly to us, but it matches the evidence. tuberculosis, you tend to become very pale, you waste away slowly, and it's like something is sucking the life out of you. It sure looks like vampires. But that is a conclusion that is not supported by the scientific method. Okay? We can, well, and we'll get to that when we talk about Koch's postulates about how Koch came to the conclusion that a bacterium, a microscopic bacterium, causes tuberculosis. Okay. All right, any questions about the difference between observations and conclusions? Okay, so it's easy to jump to a conclusion without having data to support it. Okay, we call this an inference observation confusion or jumping to conclusions. Okay, so like the folks in New England who were quite sure that vampires were stalking certain families. And, you know, that's, you know, it supported the data, but we didn't have enough data to support the inference that vampires were causing deaths of extended families. Okay. It's really easy to do. I have jumped to conclusions before, and I will in the future. Because you see an observation, you come up with a rational reason for it, and you want to run with it without collecting the data to see if your idea of what is causing the observation is correct. Okay. Okay, any questions about that? Okay, all right, let's hop over here to a lovely diagram that I have of the basic steps of the scientific method. Now I've grouped them into 3 parts. Okay, there's more than one step within each of the parts, but let's go ahead and go over them. So the first thing is you have an observation, okay, or a question. They usually happen right at the same time. Okay, I call it the stage of the scientific method. I wonder why. Hey, I see this and I wonder why. Okay. So then what we need to do next is take that observation, that question, and build an experiment with it. Okay, that to test my idea of what's causing the observation or the question. Okay, so we call this a hypothesis. A hypothesis is a testable question. Okay, so it usually has just one parameter. Okay, one thing I'm going to test, because if I'm trying to test multiple things, it gets too confusing. Okay, and so for example, okay, we could say that you know, hey, every time I get the flu shot, I get the flu. I wonder why that is. So my hypothesis could be, hey, I'm getting influenza from my flu shot. That's my hypothesis. Okay. So I would need to build an experiment to test that. Okay. So let's say I say, okay, this year I'm going to get a flu shot, and I have access to testing facilities so that I can do rapid tests for to see if I have influenza virus. Okay. And so I take nasal swabs for a week and I do rapid tests before I get the shot. Or what would be better is if I had somebody else do the test, you know, swab my nose. I get the shot for a week after, do the nasal swab, okay? And I record my symptoms along the way, okay? And so that would be the experimental design, okay? And then I would also get all of my friends who get influenza symptoms after getting the influenza shot, and I would get them to be part of my experiment. Because you can run an experiment of one person, but it's not statistically accurate. And then I would get people who get the influenza shot, but don't get influenza symptoms, okay? So that would be my hypothesis, and that would be my experimental design, okay? And then I'm going to collect and analyze the data, okay? So we all do this at the same time. Somebody collects the data, they analyze it, and we find out that, you know, the folks that nobody has virus in their nasal passages before or after the shot, okay? So, but the same people keep getting influenza-like symptoms, okay? So we analyze the data and we say, okay, it can't be influenza. Okay, maybe it's another virus. Okay, maybe it's my immune system reacting to the immune stimulation of the influenza shot. Okay, so that would be another experiment that we'd look at. Okay, so what we do is we collect and analyze the data and we say, okay, does it support my hypothesis? In this scenario, no, the data do not support my hypothesis, okay? So then we go back to, okay, we still have this observation that needs to, this question that needs to be answered. So now we're going to start over, okay? All right, questions about the scientific method and the steps of it. Now in lab, for those of you that I saw yesterday, you already know about the experiment that we're running in lab. That's what we're going to do. We're looking at, does anybody remember what the hypothesis is for the experiment that we're running in lab? And it's okay if it's a spoiler alert for those of you that haven't been in lab yet.
Toxins in sagebrush?
Very good, toxins in sagebrush, yes. Okay, so do you remember what the question is? We have the observation. Do you remember what the observation is between the two different kinds of sagebrush? It's okay if you don't remember. Repetition is the key to learning, and forgetting is a part of learning. Okay, we're going, I have no idea. I don't remember that part. All I remember are toxins and sagebrush, which is good. For a first time through, that's what we're going to do, is we're going to remember part of it, and your brain's going to go, okay, I don't need this other stuff. But if I remind you, then you'll keep it for a little bit longer, okay? So we're looking at toxins in sagebrush, okay? There's two different kinds of sagebrush, early sagebrush, which is native to Idaho, and Wyoming sagebrush, which is not, even though we're neighbors. And Wyoming sagebrush is very toxic. The animals in the area can't eat it and live, okay? So they nibble it, they get sick, they stop nibbling. So we're looking at, our hypothesis is, maybe there's some bacteria that live in the leaves that are producing the toxins. So we're going to test that by seeing if there's different bacteria inside the leaves of Wyoming sage versus early sage. Cool, right? And in a different class, biology 3, they're going to be extracting some chemicals to see if there's these bacteria are producing higher levels of toxins. In the intro micro lab, we don't have the time nor the techniques to do that. But you guys are going to be helping to isolate new fungi and new bacteria. Okay, cool, right? So we're going to be following the scientific method. We have the observation that Wyoming sage is much more toxic. Our hypothesis is that maybe it's the microbes in the leaves. Okay, so we've set up an experiment and we're going to be collecting and analyzing data, but it's going to be over the course of several semesters because we're probably going to isolate a whole bunch of bacteria and fungi. But you're going to help. Isn't that cool? All right, let's go to Koch's postulates. Okay, So Dr. Robert Koch was a German scientist who lived at the same time as Louis Pasteur. Actually, they disliked each other immensely, partially because Louis Pasteur was French, Robert Koch was German, and at the time they had just finished a war. And so they did not like each other very much. If you ever want to read interesting account of scientific rivalries, those two are very interesting. But Koch came up with what we call Koch's postulates, or Koch's postulates of disease. Okay. So he was able to definitively show, okay, that he started with anthrax. I usually use tuberculosis as an example because most people are familiar with tuberculosis. Not as many people are familiar with anthrax, even though we had anthrax spores flying through the mail service in 2001. That was kind of a long time ago. So even though he originally did this with anthrax, we're going to talk about tuberculosis because he did pretty much the same thing. Okay. Now, there is a cow form of tuberculosis, and then the human tuberculosis, you can infect mice with it and they can get tuberculosis. Okay. So in Koch's postulates, okay, he collected samples from a bunch of people that had TB, found the same huge rod-shaped acid-fast bacterium in every case of tuberculosis. Okay. So that's the first step, is that you need to find the same organism in every case of the disease. Okay. And not find it in people who don't have tuberculosis. Okay. So then what you do is you take these samples and you culture it in the lab. Okay. You put it on agar plates. Okay. Actually, at the time, he used a variety of things from potato to all sorts of other stuff. But actually, the wife of one of his lab assistants suggested, hey, why don't you use agar? Agar is something that most bacteria can't break down, so that's why we still use agar. Cool, right? So you have to culture it and get a pure culture. Okay. Then what you do is you inoculate this pure culture, just those big rod-shaped acid-fast bacteria, into a new susceptible host. Okay. You inject it into mice. Okay. And then the mice get the disease, and we're able to isolate the same big rod-shaped acid-fast bacterium from the mice. And then you do it again. Okay, you culture it, get a pure culture, inject it into more mice. The mice who were healthy before get sick. Okay, and that's how we say, okay, this big rod-shaped organism causes tuberculosis. And we've been able to do that for several pathogens. Okay. True pathogens. Okay. So when would we have to modify Koch's postulates? When would these steps not necessarily show that a particular pathogen is causing a disease?
I have a question
Sure.
about the organism in every case of the disease.
Mhm.
And is it also have to be 100% true that it is not in those that don't have it?
For Koch's postulates, yes, but do we have situations where people have that organism and they're not sick?
Yes, that's what was confusing me. So just according to
Okay.
this postulate is, if you're proving it with this, it has to not be in anybody else. They can't be like just carrying the bacteria asymptomatic.
Exactly. Exactly. That's where we have to modify it, because some people, like you say, carry it asymptomatically. Okay. So with tuberculosis, actually, that's one of them that we've had to modify, because people who don't have the disease, but it's latent, it's in their lungs, there's little tubercles They didn't have x-rays back then. Yeah, now we can do an x-ray and say, oh, you have latent tuberculosis and you might develop active tuberculosis.
Oh.
Yeah. Okay, Rochelle, you had your hand up.
Oh, I was just going to touch on what you covered on step 3. And another example would be if you knew it caused a disease, you obviously wouldn't want to infect somebody because of ethical reasons.
What? We wouldn't want to infect people with HIV virus to see if it causes AIDS?
Our government did it with syphilis back in the day. So
Oh, yes, we did. And that is so not ethical. And
Nope.
we don't do that anymore. At least we better not. So yeah, they used to do that back in the day. They would infect people with things like syphilis, smallpox. When they were developing the smallpox vaccine, they tried it out on convicted criminals, and it's like, yeah, we're going to hang you anyway. We may as well give you smallpox and see if it, cowpox, and see if it protects you from smallpox. Yes, Michelle.
How do they decide or discover what's a susceptible host?
So what they do is they try it out. With some things, like with anthrax, originally it was just a disease of cattle, cows, goats, sheep. Okay, so we knew that cows, goats, and sheep would get it, and the herders would get a very mild form, but they wouldn't pass it from human to human to human. Okay, so when Koch was figuring out anthrax, if I remember correctly, he had mice that he tried it out on to see if they would get anthrax. And as it turns out, mice can get anthrax too. So he would isolate his original cultures from cows, sheep, goats, and then inoculate it into mice. And that's generally what we do, is we try things out on mice first. They're easy to grow in the lab. They reproduce quickly, which means they die quickly. And there's fewer ethical considerations. Most people don't feel too bad about, mice living in labs. By the way, we do treat them nice. I have worked with mice and rats, and they live happy, productive lives. And they're surprised when their number comes up. So, isn't that terrible? But they've got it pretty good up until then. We even have a dating service for them. It's easier than in the wild. We don't make the males fight it out. So yeah, so that's part of how we figure out what a susceptible host is. And there's some human pathogens that will only infect humans. And so we have to modify Koch's postulates. So for example, with HIV, there are some related viruses that infect cats. And so a lot of the research on HIV has been done with feline immunodeficiency virus. Yeah. And so most people feel a little more queasy about cats, but once again, we treat them very nicely until their number comes up. And then we treat them nicely, even though we're injecting them and potentially giving them an autoimmune disease. So, okay, other questions?
Can humans contract that from the cats? If the cat has it and say they get scratched, can that transfer to humans?
I'm going to give you what I know, which is I'm not sure, but I'll ask Holly Paquette. She's another intro micro instructor, and she actually worked with feline immunodeficiency virus. So far as I know, I don't think it does. I don't think that we can get FIV from cats. But I would imagine that they are very careful while working with it, because just in case. But I'll double check with Holly and find out.
Okay, thank you.
Oh, you're quite welcome. Yes, Michelle.
Just to add on to that, I had a cat that they suspected had FIV, and they told me that I didn't need to worry about it, but that he was highly contagious to other cats
Mhm.
if they scratched each other and stuff. So I had kind of researched it back then, and unless it's changed, that was what the vet told me. After
And-
$500 worth of tests, they said he doesn't have it.
It does get expensive, yes. But yes, thank you for confirming. That's what I had understood. But you had the actual experience and whereas that's just what I read once. Not as nearly the number of repetitions that you had learning the material. So, yep. Okay, before we leave, oh, Candace,
I had a question.
sure.
My friend, I don't have a kid, but my friends all pretty much went to the same daycare.
Mhm.
And this was quite a while ago, when there was a chicken pox outbreak.
Mhm.
And so the daycare lady did the popsicle thing where they all had a popsicle at the
Ugh.
same time,
God.
so that all the kids would just get chicken pops.
Did she ask the parents first?
Yes.
Okay,
yes.
good.
So they just all got it at the same time just to get it over with. Is that kind of applied to this?
That's kind of, yes, that's kind of the at-home scientific method. I
My mom
heard
did
of
that to
the-
me.
Yeah, I've heard of chicken pox parties, which back before they got the vaccine was pretty
Yes.
standard because it's like, okay, let's just get it over with. And most people don't die from chicken pox.
No.
But and it's a whole lot safer now that we don't have smallpox circulating at the same time. Back when we had smallpox circulating at the same time, the initial symptoms are the same. And so parents would think that their kids had chicken pox, which is a fairly mild disease, and they'd have a chicken pox party. And it turned out it was smallpox, and a quarter of the kids died.
Oh my goodness. Okay.
That's
Yeah. So if you're sure it's chicken pox, which we don't have smallpox circulating, and it's probably, as long as all the parents are okay with it. Maybe the parents don't want to deal with 3 kids with chicken pox all at the same time. They want to space them out. Get one sick, get them better, get one sick, get them better. Let's see, Rochelle.
Yeah, I have a question while we're on the subject of chicken pox. If you never had it, but you were vaccinated, what does that do for you when you're older and you get shingles? Are you going to get it worse? Are you going to get it less? I've heard a lot of mixed things.
Yeah, I've heard a lot of mixed things, and I think part of the problem is the chicken pox vaccine has not been around for that long. And so the folks who were vaccinated are just getting to an age where they're likely to get shingles. Okay, so because we don't have that many people who have had the vaccines getting shingles yet, that's why we're having mixed results. Personally, because I've known people who have had mild cases of shingles, and I had a friend who got horrible, weird shingles that instead of being external, it was internal. It went up his auditory nerve, and he was temporarily deaf for a while, and it went down his vagus nerve, and he couldn't swallow. Ended up in the hospital for a month while they were trying to figure out what in the world was going on, because it's very unusual shingles. It usually doesn't happen that way. And he ended up contracting an autoimmune disease because he had his shingles inside and his immune system went crazy and started attacking his muscles, and he eventually passed away from that. But like I said, that's very, very rare. Most people get it, and it's highly painful, highly contagious. And you can't get shingles from somebody who has shingles, But if you've never had chicken pox and you haven't been vaccinated, you can get chicken pox as an adult, and it's very severe and it can be life-threatening. So personally, I would recommend getting the shingles vaccine, which is basically a booster to up your immunity about the time that your immunity is dropping off. Yeah, you don't want to get chicken pox as an adult. If you can avoid shingles, avoid shingles. Yeah.
Just to clarify on that, they are different strains of the same virus, correct?
No, it's the same virus. It's just where it's hiding out. So
Gotcha.
tell you what, let's go to the whiteboard. Because this is one of these that I did my master's research on herpes virus. So herpes is kind of my thing. It's not something that anybody enjoys getting, but it's interesting to study. Okay, so here's my person. Okay. Well, let's do it. I'm going to pre-do my cartoons. And yes, my cartoons are horrible. And then I'm going to have a kid. Okay. Okay, back in the day before the vaccine, most of us became infected with chickenpox, which is unusual for herpes virus because it's respiratory. Okay. Gets into your respiratory and then proceeds to go all over your body, and it's really, really itchy. I'm old enough that we did not have a chickenpox vaccine. And if you scratch it, you can become infected with a secondary infection of staph, and it causes, scars and what have you. So what happens is your immune system sees this virus that's all over, and it attacks, and you get better. But that virus is still there. Instead of being on your skin causing lesions, these fluid-filled blisters that we call a pox, it has gone into hiding, and for most of us, it goes into hiding in the nerves along your spinal column that supply the nerves to your trunk, your chest, and your back. Okay. And they hang out there. Your immune system can't see it. Okay. So, but you have antibodies. So when we breathe in more chickenpox virus, you're immune, your antibodies take care of it. Okay. The vaccine does the same thing. Okay. Well, what happens as we get older, everything falls apart. And your immunity to viruses that you got as kids drops off. Okay. So what happens is, and I should have given this person a trunk, but as the antibodies to the virus drop, okay, then the viruses that have been hiding around in your nerves come out, and they form lesions, usually in a very limited area on your chest or your back, or in a friend of mine on his elbow. Okay. Just depends upon where the virus is hanging out. It's the same virus. It's just been hanging out, hiding in your nerves, hiding from your immune system. We call this latency. Okay. If we were to swab your mouth, your nose, when you have chickenpox, we swab the lesions. We can pull virus out of those, okay, and culture them. When you're in latency, we're not going to go in and sample your nerves. That would be too painful. Okay. So we call it latency, but we swab your nose, you don't have any lesions, there's nothing in your blood, so it's hiding. Okay. But when it reactivates, okay, that's what we call shingles. So it's the same virus, it's just how it presents. Chickenpox is all over your body, shingles is in a limited area, and it's a reactivation of latency. Yeah. So did that answer your question, Rochelle?
Yeah, I have one more for you though. You
Sure.
say shingles, well, the virus that kind of hides out in the nerves. If they were to do like a test with your cerebral fluid, could they see it from that kind of nerve tissue? No.
Nope. No, because it's inside the nerve and it's not replicating and it's just hanging out. It's not doing any damage to the nerves. It's just hiding. Yeah. And so the shingles vaccine is a booster to up your antibodies so that when they do start to move out, your antibodies take them out before you get shingles. Yeah, all
What-
herpes viruses do that. Oh, sorry, I just dropped my electronic pen. Yes, we had a question.
What causes it to reactivate?
With depending upon the herpes virus, with shingles, it's a drop in your antibody levels. It's a suppression of your immune system. And so it's, think of it this way, they're in hiding. There's a group of them, but occasionally they send a scout out to see if your immune system is not looking. And if they don't come back, they say, okay, we're going to stay in hiding. But as we get older, our immune system decreases. Sorry, everything decreases as you get older. And so they send scouts out and they don't die. And so the others just keep coming out and you get shingles. But if you get a shingles vaccine, that gives you a booster that ups your antibody levels and you're highly unlikely to get shingles. In biology, nothing is 100%, but it decreases your chances significantly. So interesting. And this is another one where Koch would not have been able to figure this one out. Yeah, but it's the same virus, and that's why they tell you you can't get chickenpox more than once, because you still have it. You don't get better from it and then get it again like you do with influenza. Yeah. Okay, questions? Okay, let's see. We have 5 minutes left. Let's go back to the PowerPoint. By the way, good questions. I just have to do some fairly tight pacing. Oh, no, we don't have 5 minutes left. Never mind. I misread my timer there. We've got plenty of time. We've got another half hour. Okay, so microscopy. Okay. Microscopes are what we use to figure out, to look at microbes. Okay. And there's 3 of them that I want you to know. Okay. One of them is bright field. So that's one where we shine a light through a specimen, and we can look at things to a fairly small size, but light has a limit. Okay, actually, let's go back to the whiteboard and I'll explain briefly how light works. Okay, so if I've got a light microscope, okay, I've got what we call an objective, okay, and then over here, I've got an eyepiece, and that's the part that you look through, okay, and we're going to do blue just because my eyes are blue. Isn't that a cute eyeball? It's kind of a scary eyeball. Okay, so you look through the eyepiece, okay, and down here we've got our specimen, okay, on a slide, and I'm drawing it huge, okay, but I've got some little bacteria on here, okay, and I have a bulb here, drawing the old style Edison style bulb, and so we shine light through it and it bounces around, okay, when it hits, okay, and it comes in here, we concentrate it, we make it bigger, and we send it into my my eye, and they look big enough for me to see, okay. So that's a bright field microscope, okay. Part of the problem with the bright field microscope is that one, most organisms are clear. And so we have to stain them. Okay, if I've got algae, they've already got their own pigment. I don't have to stain them. But say I've got a bacterium that is not photosynthetic, I have to put a stain on there to be able to see it. And generally speaking, the stain kills it. Okay. So there's another microscope that we call a phase contrast, and we mess with these bending light waves so that we don't have to stain them. What we do is we get shadows. Okay. And so that enables us to see the organism better. Okay, so if I've got something like this and I've got a nucleus, I get shadows that make those much more obvious. And I've got some little cilia and I get shadows so that I can see the cilia better. And we call that a phase contrast microscope. Okay, and then we have electron microscopes. Okay, electrons can see can help us see much smaller things. Okay. So they're set up kind of the same way as this, only upside down. And instead of shooting light, we shoot electrons, okay, through there and do kind of the same thing. And we can't see electrons like we can see light waves. So let me go ahead and draw an electron microscope. Generally, we have our electron source up here. I've got my sample down here, okay. So we'll say I've got bacteria here. Okay, and I've got my detection unit here, okay, and then I've got a screen up here that I can see. So we shoot electrons through there. The electrons come through, they bounce, they show up on the detector, a signal comes over here, and I look at the screen, okay. Whoops, I've got my eye going the wrong direction. Where's my eraser? There's my eraser. Okay, and so I can see what's on there, okay, and I can see the bacterium really big. Okay, and better details than if I were using a light microscope. Okay. All right, now we're ready to ask questions. Okay, so I'm going to pull up a poll that has 3 multiple choice answers. And my hope is that you did some reading on chapter 1. And so you've, my very brief going over the microscopes, okay, is a review. And it's okay if you guess. Okay. All right, so in looking at this and looking at the size scale, was this picture taken using a bright field microscope, an electron microscope, or a phase contrast? And we've got some early voting. Good job. And with practice, it's okay if you're wrong. We got some good voting going on. Okay, we've got about 88%. Ooh, now we got 92%. There we go. We got 96. See if we can hit 100. I think we stalled. Okay, so here are the results. Okay, we were split between electron and phase contrast. Okay, and then we had a few people pick bright field. Okay, who would like to tell us why they picked what they picked?
I picked B because it had a dark background.
Okay, and that is actually one of the best ways to determine. Okay, So that is a good valid background, good valid way to pick. Okay. So which one would definitely not have a dark background?
The bright
Right.
field.
Very good. Bright field has a bright white background. Okay. By the way, if you pick bright field, that's okay. Like I said, we're practicing. And you probably didn't know that. Okay. Let's see. Amanda.
So in the book, there's a dark field which looks very similar to an electron one. So would they be the same or different?
Dark field is like a bright field, but they've messed with the wavelengths so that everything is in shadow. So the way to tell the difference is by the size. So this is pretty small. Okay. A micrometer is a millionth of a meter, and a meter is about a yard. Okay. So this is itty bitty, itty bitty. Okay. So one way to tell the difference between a phase contrast or a dark field and a bright, because they both have dark backgrounds, and an electron microscope is by the size scale. If we were talking about 10 to 40 micrometers, that's in the light microscope range. If it's smaller than that, it's going to be electron. So dark field, how do we tell the difference between dark field and electron microscopes? By the size, by the size. Also, and this is going to be, oh dear, why didn't you tell me that to start out with? Electron micrographs tend to come out in black and white. So we put in what's called false color. These bacteria are not actually orange. We went in and added color. We also darkened the background to give better contrast. But when you look at these guys, look at all that fine detail. That's like you're sitting right next to them, just as small as they are. And that's also one way you can tell an electron microscope. Okay. Okay, you want to do another one? Let's do another one. Okay, here's another one. I'm going to stop sharing and I'm going to relaunch this. Okay, for this one, bright field, electron, or phase contrast? Oh, got some good early voting. Ooh, we just passed 50%. And we passed 75%. Okay, we're stalled at 92. Oh, there we go, 96%. Okay, most people picked phase contrast. Okay, next most favorite answer was bright field, and then we had a little bit of voting for electron micrograph. Okay, okay, who wants to tell me why they picked what they picked? Hey, Logan.
So my guess is phase contrast, simply because we can see the villi of the cell itself, the actual structures that you normally wouldn't be able to see in the bright field microscope.
Right, very good. You are correct. In a bright field micrograph, this would look flat because it doesn't have the shadows. See those nice shadows? Okay, it's almost like those pictures that we take of the surface of the moon or Mars. Okay, kind of the same thing. You get the shadows and it makes it 3D. Okay. Okay, if you picked bright field, you were close because phase contrast is a light microscope. Okay. And if you picked electron, don't worry. I'm sure you picked it because of all this nice detail, but this is way too big. Way too big, yeah. And this is the actual color that we see from a phase contrast. They tend to all be like black and white or this lovely blue overall tone. Okay. All right, are you ready for one more? Let's do one more. At least on this topic. Okay, here's another one. Okay, is it bright field electron or phase contrast? By the way, these are blood cells, and these are trypanosomes, which I wouldn't want to get any of them, but man, they're cute. They look like little eels swimming around in this poor person's blood. Okay, we are at 92%. I need just one more. There we go. All right. Oh, we got to 100%. Woohoo. Okay, most people picked bright field. We had a few people pick electron or phase contrast. Okay, who would like to tell me why they picked what they picked?
I picked bright field just because the white contrast in the background.
Exactly. It's got a white background. Okay. Also, they're looking pretty squished. Bright field tends to look squished. And also, we had to stain it. You wouldn't know that, but all of these cells originally were clear and we had to do a stain. Okay. All right. We feel pretty good if on the test, if I gave you a picture, would you feel pretty good about picking out the right one? And I would give you a choice between these three. Yeah, and flip through your book. Your book will tell you whether it's a bright field, electron, phase contrast, dark field. Okay. Candace, do you have a question? See your hands up. It takes a bit to get to the mute. Okay, if you do, go ahead and let me know. Oh, I have another one. Let's see, how are we on time? Oh, we've got time. I said we weren't going to do another one, but let's do another one. I forgot I had 4. Okay, this one, is it bright field electron or phase contrast? Okay, we've got some good voting. We're at 96%. Hey, we're going to call that good. Now, this one, we had even picking between electron and phase contrast. Okay, and part of it is I oversimplified things, and I think this was throwing you off. Okay. Okay, who wants to tell me, somebody who picked electron and somebody who picked phase contrast, tell me why you picked, and it's okay if you got it wrong. I'm going to help you figure out between the two, even when the scale throws you off. Okay, Lily.
I chose phase contrast because of the size.
Because of the size, because I told you if it wasn't itty bitty, then it wasn't going to be phase contrast. It wasn't going to be electron. Yeah. Okay, Logan.
So I chose electron because we are able to see the outside and none of the inside structure of this microbe. And that's how electron microscopes work.
Yeah. Okay, you both used good reasoning. I had forgotten that, yes, sometimes we do use electron microscopes for relatively big things. So that was my fault. I threw you off. Okay. So Amanda, yes.
So I picked phase contrast, one because of the size and because of how detailed it is. But then I realized after I answered it, I was possibly wrong because of the background, because it seems like now for phase contrast, is the background also more detailed as well?
Uh-huh. You are correct. Yes, if we go back, let's go back to that phase contrast. Oh, let's see. Come on. Let's see. I've got to go back. See all the other stuff? We've got little bacteria along with this protozoan. Okay. Generally speaking, in electron micrographs, and this isn't all the time. Okay, it depends upon what they're taking a look at. But you're right. Most of the time, we're not going to see that community. Most of the time. Sometimes they do. But like this one, they were looking at just this protozoan. Okay. Also, when you see color, we generally do not put color on phase contrast. Okay. And look at all that fine detail. Oh, isn't that lovely? We see these little pore-like looking things. We've got the paramecium's mouth. We've got all these cilia. Okay, it's very, very detailed. And even though the phase contrast is detailed, look at all these shadows. We're getting the detail from the shadows. So we've got a shadow here that makes the cilia pop out. And it's because of the shadows. Here we're looking a little more flat because the light's coming from this direction. Okay. So see the difference between the two? Yeah. Okay, so no matter what you put, you did a good job because you were going with the information that you had. And my apologies for telling you that we never did electron microscopes with big stuff. I was wrong. Sorry about that. Okay. I think that's the last one. Let me double check to make sure oh, yep, that is the last one.
I have a quick question.
Yes.
So I had to step away from my computer for a second. So what was
sure.
the answer on that one?
The answer on that one is, let me back it up if I can. Is electron microscope.
Okay, thank you.
And Amanda, sorry, I missed that your hand was up.
Sorry, I forgot to lower my hand.
Oh, no worries. I do that in meetings all the time because up until about this year with my newest update on Zoom, after a certain amount of time, your hand would go down by itself. Now you have to click on the thing to lower it. And so I do that all the time too. Oh, and I forgot there was a question I was going to ask. So thank you, Alexa, for having us go back. Which microscope are you going to be using in our lab? Bright field, electron, or phase contrast?
Isn't it going to be bright field?
It's going to be bright field, yes. Bright field are less expensive. They're also a whole lot tougher. You can use them for years and years with multiple people before they break down. Electron microscopes are huge, expensive. You have to have them on a cement pad to decrease movement. Yeah, we don't let students use them. Phase contrast, kind of the same thing. They're a little bit more expensive and they're a little bit more finicky. So bright field microscopes are the workhorses of the micro lab. We generally start out in any lab with the bright field because it's just a good place to start. You get a good look at it and then you move on to the more advanced stuff later. Okay, let's talk about gram stain. Okay, this chapter talked about staining, and we haven't gotten to that lab yet, but this is a good preview. And so you'll know more about it for when you actually do it in lab. Woohoo! Okay, so here I have a gram-positive rod cartoon, okay, gram-positive bacteria. And here I've got a circle-shaped bacteria, okay? By the way, we have specific terms for these. If it's rod-shaped, it's bacillus. If it's more than one, bacillus if it's one, bacilli if it's more than one. Circles are cocci if there's more than one, coccus if there's one, okay? So I've got a gram-positive bacillus here that I'm going to stain, and I've got a gram-negative coccus that I'm going to stain, okay? And they all start out clear, okay? So I've got to stain them. So I'm going to do the gram stain, which is called a differential stain, because we can differentiate between different types of bacteria based on the stain, okay? Then it all goes to the structure of the cell wall, okay? And we'll learn more about the structure of the cell wall in chapter 3, which is coming up, okay? So the first thing I'm going to do is I'm going to put a purplish blue stain on called crystal violet, okay? So I'm going to put that on, and that's going to stain both kinds of cells purple, okay? At least to me, it looks purple. Other people, it looks blue, purple, blue. It's kind of in the same family, okay? So then I rinse that off. Then I put on iodine. Iodine is a brown color, okay? If you've ever put iodine on a cut, you know, it's kind of a tannish brown. And that doesn't change the color, but it acts as what we call a mordant. It sets the stain and makes it harder to get rid of in the next step, okay? So it binds to the crystals of crystal violet and it makes the crystals bigger, okay? So I put iodine on and I rinse it off. Then I do my decolorization step. In the book, it talks about acid alcohol. We don't use acid alcohol. I don't know anybody who uses acid alcohol anymore. That's what Graham, who is a student of Koch, used to use. Now we use 95% ethanol, okay? So what that does, our decolorizer, the gram-positive cells, because they have a thicker cell wall, are going to keep that blue crystal violet stain, okay? The decolorizer is going to take the stain off of the gram-negative cell, okay? They've got a much thinner cell wall, and it's going to wash the stain off, okay? So at this step, my gram-positive is still purple. My gram-negative has now become clear. Well, clear on a white background is really hard to see. So we do what's called a secondary stain. We use safranin, which is red. Now the red doesn't, you know, I've already been stained with purple. It's a darker stain, okay? And so after the secondary stain, my gram-positive are going to be purple and my gram-negative are going, I see red. The book says pink, red, pink, they're in the same family, okay? So they're going to show up as either red or pink, okay? Okay, questions before I start asking you questions. Okay, so I've given you the model, how things are supposed to go. And now I'm going to tell you, hey, what if we changed this? Okay, so you performed a gram stain, but forgot to add the decolorizer. What would the gram-positive bacterium look like underneath the microscope? Well, let me, come on. Oh, let's see. I need to switch my poll. Okay, how do I switch my poll? I want a different poll. That's interesting. There we go. I don't know what I did, but now I'm able to give you 5 choices. There we go. So remember, I'm asking about what a gram-positive bacterium would look like if you forgot to do the decolorizer. And it's okay if you get it wrong. This is part of study, is testing what you remember. And if you don't remember, that's fine. You need more repetitions to really remember it. Okay, we're at 72, 76%. Okay, I'm at 88%. Let's see if we can break into the 90s. Okay, we're at 96. For the sake of time, I'm going to end the poll. Okay, so most people picked blue purple. Okay, a few people picked clear colorless, and a few picked brown tan and pink red. Okay, all right, who wants to tell us what they picked and why? Yes, Amanda.
I picked B, the blue purple, because even if you skip the decolorizer, it was purple before, so it would still be purple afterwards.
Okay, very good. And you are correct. Okay, that is the correct answer. If you picked clear colorless, brown tan, or pink red, don't feel bad. Like I said, you've got to have some repetitions before it really sticks. Okay. Now I'm going to ask a follow-up question. What would a gram-negative bacterium look like if you forgot to add the decolorizer? So I'm going to relaunch this. Yeah, let's relaunch the poll. Oh, whoops. I ended the poll. Whoops, come on. Okay, I clicked the wrong thing, so I'm going to have to relaunch the poll. There we go. Yeah, we're got some good participation. Good job. We just broke 70%. Ooh, we just broke 90. There we go. Okay, let's see. Let's go with Amanda first and then Michelle.
Well, I picked blue purple again because same reasoning as before. Because it was blue purple before that step, I would assume it would bleed blue purple after.
Okay, very good. Okay, Michelle.
Oh, I think I messed this one up. I thought it started out red. So I pick red, but my question is that what if you haven't used the decolorize or done that step, what does the saffron do to the slides? Does it just have no effect whatsoever because it hasn't been decolorized?
Yep.
Okay, so
yeah, because they're already stained with that dark purple stain, and the red stain is much lighter. Besides, it's already stained.
Okay.
Yeah. And getting it backwards is just fine at this point. When you're first learning a concept, it's easy to get it backwards as your brain is trying to figure out, my analogy is figure out what shelf to put it on. And so that is perfectly okay that you got it backwards. That is normal. Okay. But the correct answer is, yeah, it's still going to be purple. They're all going to be purple. Yeah. Okay. So we're pretty close to the end here. So this is a good point to stop and say, okay, what we're going to do for next time. Okay. So here are the assignments that are due on, okay, I completely got that wrong. These are not the things that are due on Friday. Let me stop share and pull up the schedule because I need to fix the PowerPoint. Sorry about that. Okay, so things that are due. Okay, you can turn these in this week, and I won't take points off. Okay, but I do want you to participate in the icebreaker discussion board. That gives you guys a chance to talk to each other and get to know each other, even though you're not actually talking, you're typing. Okay, so I want you to submit an introductory paragraph about yourself. Supply a picture if you're comfortable doing that. And then I want you to respond to at least 3 of your classmates and say, hi, or hey, I'm in the same major, or I like hiking too. Okay. And then we have the honor lock practice exam. Okay. So all of our tests will be proctored through honor lock. And so I want you to try it out, and we can work through any technical difficulties before the exam in 3 weeks. Okay, that gives us time to work out all of those technical difficulties. And then I want you to do the chapter 1 practice quiz. This is the only practice quiz I'm going to have you do. But I want you to do at least one so you can see that it's available. The questions were written by me, and they are examples of the harder style of questions I'm going to ask on the exam. So you can see how my mind works. And you can take it as many times as you want and until you get full points. But I would treat it like a real quiz, you know, closed book, draw out the problem to work your way through it, that kind of thing. Okay. All right. Questions about what's due on Friday?
I had a question.
Yes.
So if the library badge is due next week, shouldn't we work on that first instead of the biochemistry badge? Or does the biochemistry badge take longer and that's why you want us to start on it first?
So you are correct. I would work on the library badge first. And then once you do that, once you start on, once you do that, then you can start these because they are going to take you longer, but they're not due.
Okay.
But yeah, the biochemistry badge, it's several modules, and most students take between 3 to 4 weeks to finish it. So if you do 1 or 2 modules per week, that's a pretty good pace. Yeah. So good question. Thank you, Elena. Okay. All right, let's go back to the PowerPoint, even though it is wrong. Whoops, I didn't mean to stop share. Okay, suggestions for preparing for lecture next week. Okay, I would start chapter 2 in Sarago, and then read chapter 2 in the textbook, or you can go to the study aids and watch the chapter 2 videos, and then write down any questions you have. So do the assignments first, then move on to the week 3 lecture stuff. Okay, any questions? Okay, I have a class at 11:30, but I have office hours after that, starting at 1:30. So if you do have questions, feel free to ask. I'm going to go ahead and